All right, good morning, everybody. Sorry for being a couple of minutes late. Dog walk went a little bit longer than intended. Hey, love, I've measured the kibble out properly, okay? Okay, love. Okay, where are we at? Doop-a-doop-a-doo. Sorry, not very organized today for some reason. It was crazy because I got up really, really early too. Okie dokie. So we were kind of like on the last stretch, really, right? This is the last section, and we've got two big topics to cover. And they're going to be tough ones. Basically, is kind of the long and short of it. And so we've got these two to cover. These are going to take at least actually, when is this? Probably up until Thanksgiving, whenever that is. Oh no, Thanksgiving is pretty much the end of the course, which is kind of crazy. Really weird how that happens in terms of the semester schedule. Anyway, so pretty much we'll be done by Thanksgiving. Maybe we've got a little bit of extra time after for catch up, and our last class and exam will be on the 1st of December. And so that's last day of classes. It's not a final exam. It's not cumulative in the traditional sense that you're probably familiar with. It's just over this section, but there is a lot of stuff going on in this section. And so the two biggest parts are these ones. Oop, that didn't work very well. Basically, the control of gene expression in prokaryotes and eukaryotes. And then we have a bunch of kind of fun stuff to talk about, which is sort of up in the air at the moment. I've got some ideas and things to change and stuff. So that's a little bit kind of make it up as you go kind of thing, really. Just because there's lots of fun stuff to talk about, and I have to make sure I have some kind of coherent plan to talk about it. So it's not just a collection of woo, which is kind of fun, but you know, I want to be a purpose to it all. So there's this one, right? And that's Mahusive. And there is this one with the famous Mr. Tuxedo toe beams. This one is also huge. Okay, so I always kind of struggle a little bit. Let's get rid of these. So we've got to free up a little bit of system resources. Oh, and also, unfortunately, I need to leave a little early today because I have to take my daughter for an MRI. And so the only time they could get us in this week was kind of 5 minutes after the end of class. So I have to leave a bit early to get there on time. Okay, so let's get back to here. So essentially, the big deal with this whole section is that really I'm going to move off from that because that's a bit of a scary slide. The DNA is just the information. The proteins typically, or RNAs, depending on what kind of gene we're talking about, are the things that do stuff. Now, gene expression as a whole, producing the mRNA and then protein and then whatever else needs to do into the proteins, is extremely costly. In fact, it's so costly that when proteins cease to function or are no longer needed anymore, or even mRNAs, they're broken back down into their constituent parts and reused. In fact, if you stress cells, you kind of limit their nutrients and, other kinds of cellular stress, they'll typically start to eat themselves. It's a process called autophagy, which means self-eating, literally, so that they can kind of survive by breaking down parts of themselves to reuse, right? So gene expression isn't something you want to do unnecessarily. But because the products of gene expression are the things that do stuff, to be successful as an organism, whether you consist of one cell or millions, you have to be able to produce those products as and when they're needed, right? And so there's kind of external stimuli, right? Basically signals that a cell or bacterium receives from outside of itself, right? So has my environment changed? Am I being told to do something different, right? Has my nutrient availability changed? Has a temperature changed? Has something changed, right? Which I need to respond to as a cell. Now, again, this could be a cell as in, you know, a bacterium in your gut, which is like, don't like this no more, you know, got to do something about it. And obviously they can't move because they've got bacterium. It's not like they're going to go to your stomach or your lungs or something. They're stuck there, right? So they have to change what they express in terms of gene products, if they're capable of doing so, right? To be able to adapt to that change. Could also be, you know, you're a gut cell, right? And you need to suddenly get more water out of the gut, right? You need to reclaim some of that water so that, you know, you maintain homeostasis, right? So how do you do that, right? You know, you need to change your gene expression profile. You need to express particular genes, say like aquaporins or sodium channels or something, right, that allows you to reclaim more of that water. So those are external kind of triggers. There are also a lot of internal triggers or stimuli, right? You know, has particular levels of a nutrient or metabolite, right, which is probably a better word, right? Because metabolite could be pretty much anything. Has that changed, right? Have I produced a lot of proteins as a cell which have used up my sources of, I don't know, threonine, let's say? And if I'm able to make threonine, then I need to start expressing the genes that are responsible for turning whatever substrate threonine is made out of, and I've no idea, into that. Crazy dogs. So there are lots and lots of kind of questions. There's lots of inputs, and it's a very dynamic process. And that's the same whether you're a bacterium or a single cellular eukaryote, like a yeast, or a multicellular organism like us, right? Each and every one of the cells in your body, right, has to be able to change its transcriptional profile depending on the context and the environment and, you know, whatever's going on, right? And it's kind of weird to think about your body being an environment, but it is, right? It's a very complex environment full of all kinds of different niches, right? You know, the environment in your bloodstream is very, very different from the environment in your muscles, for example. You know, your liver is a somewhat hellish place, to be honest. If one were to, you know, pick places to hang out, the liver wouldn't be it, right? It's full of, it's where you detoxify kind of pretty much all of the nastiness that either gets put into your body, like alcohol, not like I'm a teetotaler or anything, but, you know, ethanol is a poison, or ones you generate yourself, right? Such as, I don't know, urea, actually. Ammonia would probably be the best example, right? Which gets turned to urea and then you pee it out via your kidneys, right? So sorry. Damn. Right, so the liver in particular is a crazily transcriptionally active place, right? It does an awful lot of stuff, Sorry, I didn't have the chat. Oh, thank you. There's only 2 there, which is kind of crazy. They usually go in 3s for some reason. Anyway. Also, how will the different parts of that process occur, right? So we've talked about the overall process of gene expression, of turning on transcription, generating a transcript, processing it if you're a eukaryote, and then translating it, and that translated product, that protein, being folded, modified, and localized in different places, right? And so we're going to take that kind of like basic structure and we're going to really expand it, right? We're going to talk a lot about, particularly in prokaryotes, control of transcription, because that's really where most of the exciting stuff happens for prokaryotes. And for eukaryotes, it's even bigger, right? Stuff happens at every single step along the way, right? So it's a phenomenally complex process. And in many ways, it's like one of those crazy arcane calculations or formula, right? Which has lots of symbols and stuff in it. And at the end, that's your level of protein produced, essentially, right? So there are all these different inputs, all these different steps, all of which can be modified and tweaked in some way, right? And that's one of the many reasons why this is such a complex topic. And one thing to do, which I always try and teach my students, is keep in mind the bigger picture. Why is this organism doing this? Why does it need to control the expression of this particular gene or set of genes in this way, right? What's the bigger picture? Because as you can see from this, right, there's a lot of stuff going on, right? There's a lot of details and it's very easy to get lost and have all of this stuff. Even if you're able to cram it into your head, it's just like this kind of floating soup of stuff. Actually, one of my favorite analogies, I don't know if any of you have ever watched Harry Potter. Probably most of you have. I think it was is it the Chamber of Secrets? Where there's that mass of keys flying around that Harry has to catch the correct one to be able to open the door to the next stage, whatever that was, right? All of this stuff can end up being that massive keys flying around in a room, which is your head. And you're like, which bit is the one that I want, right? And so keep that in mind, right? There's a lot of stuff going on here. Try and learn the principles, try and learn the concepts, and then flesh them out with these details. So those concepts make sense. That's really, well, at least for me, that's kind of how I do stuff like this. But I'd suggest giving it a shot for you lot too. Okay, so these are kind of some of the questions, and those questions vary obviously depending on the cell, depending on the organism, depending on the gene, right? And so there are lots of different spots or kind of levels at which regulation can occur, right? And so in prokaryotes, it's mostly, yeah, I'd probably say almost entirely at the level of transcriptional regulation. Go on my fingers. I don't know what that dog's doing upstairs. And they do have some very elegant ways of doing so. I'm always actually, every time I teach this, and I've taught this topic a lot of times, right? I'm always struck by how elegant this process is, right? Really, and in many ways, that elegance is the result of natural selection, right? So the simplest, most kind of another cool word, parsimonious way of doing this is the one that gets selected, Eukaryotes do that too, but a whole bunch of other stuff as well. So an awful lot of eukaryotic control of gene regulation is actually after gene expression has occurred in the conventional sense, right? An awful lot of it occurs in terms of how you modify those proteins, where they end up, what stuff you add to them, what stuff you take away from them too, right? And we've got a couple of cool examples of that. And then there's everything in between as well, right? So again, this is a complex topic. And so we're going to start with some basic principles, right? We'll probably get through most of these today. And essentially, these are some kind of rules of thumb, right, to work by, some language as well that we're going to be using. And so the general rule of thumb is such. We're going to talk about the defaults, and the defaults matter because how you control it depends on what its natural state is, right, in terms of gene expression. So in general, gene expression is, on by default in bacteria. Oh, so much nice to type that in prokaryotes. So to control gene expression, you need to turn it off. In prokaryotes, dang it, prokaryotes on the mind. In eukaryotes, gene expression is off by default. Right, so what do we mean by that? Essentially, what that means is that in the absence of anything else in bacteria, as long as the transcriptional machinery is present, i.e. RNA polymerase, you know, the sigma factors, stuff like that, you will get gene expression, right? So if you think about it kind of big picture, every gene in bacteria can be on, right? And obviously you don't want them all on, right? So a whole bunch of them are turned off, right? So the active process is to turn off the gene, you know, to add stuff in terms of proteins or whatever that turns off the gene. Now in eukaryotes, the opposite is the case. In eukaryotes, we have this whole genome, tens of thousands of genes, and we actually have to actively turn them on. It's like having a library full of books, and you have to go and find which cell and which book and open it at the correct page to be able to get that information out. Right. Hopefully that makes sense. So you have to actively turn it on, even in the presence of all of the transcriptional machinery. In eukaryotes, you will not get transcription. You need to add stuff to turn it on. And so to move on from that, we then need to think about the outcomes, right? And so in terms of regulation, in negative regulation, right, here's the kind of language that we're going to use. The outcome is negative, right? So negative really talks about the outcome, right? The kind of what you're doing to it. And so negative regulation, you're turning something off. You're having a negative effect on transcription. So if you turn that on its head, by default, transcription is on because you have to turn it off, right? How does that make sense? Okay. This is definitely kind of like a building process because we're going to build on these terms, and there's extra, you know, jazz on top of that, which, you know, it's kind of like a layer cake, I guess. Actually, there's a film called that, I think. I'm not sure if I watched it or not. Now, in positive regulation, again, you think about what's the outcome of that. And in this case, the outcome is something positive, right? So by default, gene expression is off, right? And so there must be things which carry out that regulation, right? So if we're turning something off, typically in bacteria, although we can have these in eukaryotes as well, we need a repressor protein, right? Something that can turn that gene off. And that's typically a protein which will bind the DNA of the gene, right? And the promoter, essentially, or in bacteria, is called an operator, which is kind of similar, and block transcription. It's actually physical blocking of transcription. Now, in positive regulation, because we're turning something on, we're activating something, right? And so we need a protein to physically bind to be able to activate transcription, right? So now we're using a transcriptional activator protein to turn transcription on. Again, remember, actually, crap, I shouldn't have shut that, should I? Here's me and my organizational skills. Where are we at? This is what I want. Oh, where are we at? It's in here somewhere. Here we go. And so again, in eukaryotes, you could have all of this stuff bound, right? All of the transcriptional machinery. Oops, RNA polymerase, all the transcription factors, so on and so forth. But without these transcriptional activator proteins, you will not get transcription, right? And so those have to be present. And that's a big part of the cell specificity of transcription, right? Why you have certain things expressed in one cell and not in another cell, right? So obviously the requirements of gene expression in your eyeballs are very, very different from in your pancreas, right? And so these transcriptional activator proteins are a big part of that differentiation, I guess, or differences. Got to be careful. Differentiation actually means something else. Okay, so we have negative regulation, turning something off that is by default on. Positive regulation, which turns something on, which is by default off, right? So keep those kind of ideas in your head. You ready to go? Okay. Uh-oh. Ah, there we go. Come back again. And so, come on, get off of it. So I got a notification up. I was futzing around my headphone. So in terms of positive regulation, this is kind of what it looks like, right? So in the absence of that activator, you don't get any transcription, right? The cupboard is bare, so to speak. Only when that protein binds to that site do you get transcription. And this protein, it doesn't bind to any binding site. It has a very specific DNA sequence that it will recognize, right? And so only if that DNA sequence is present in the promoter of that gene will this protein bind to it and activate transcription. Okay, so now we have like a couple of extra layers, a couple of extra kind of wrinkles to add to that. So now we have inducible, right? And so again, we're thinking about what is the outcome, right, of this action. And so if we're inducible, we're inducing something to happen, right? And the typical state of transcription is off due to the repressor. So we're inducing that repressor to leave, right? Going, come on, off you go. Right? And that releases the repression of that gene and you get transcription. So here's the kind of complex kind of part. It's like a logic kind of puzzle, essentially. So in, what was the exact term that we used? There you go, negative regulation. Probably consistent. So negative regulation transcription is on by default. So must be turned off by the presence, let's capitalize that, presence of an or a repressor. Oh, that's supposed to be by, not my. Sorry, type an error. Now, when that repressor is bound, transcription is off. And the presence of, oh, that's terrible, of an inducer can induce or persuade, I guess, that repressor to leave equals transcription on. So you have to kind of follow through like all the different like logic gates, really, right? It's very much a logic kind of puzzle in a sense. And so in inducible transcription, transcription is off due to the presence of a repressor. And that repressor must be induced to leave for transcription to occur, right? So essentially what you're doing is taking everything away from the DNA, from the gene, which allows it to go back to its default on state. How does that work? What do you think? Let that settle in for a little bit. Any questions, clarifications, rephrasings? Correct. Well, then you look at what needs to happen for that gene to be expressed, right? So, for example, in eukaryotes, gene expression is always positive, right? So actually in eukaryotes, it's a lot simpler to talk about, to be honest, because it's almost always just positive, right? However, in bacteria, then we have, because the default state is always on, then we have to talk about whether we're turning it off by adding something, right, which is a repressible transcription, or if we're turning it off by removing something, and that's inducible transcription. So these terms are much more useful when talking about control of prokaryotic gene expression. They don't apply really quite as well to eukaryotes. Does that help? Okay. It'll make a lot more sense when we actually talk about specific examples, to be honest. Yeah, it depends on the outcome, right? So you always have to kind of think at what's happening at the very end, right? Do we have expression or do we not have expression? If we have expression, why do we have expression? Right? Do we have expression because there's just nothing there and it's a bacteria, so it's always going to work? Do we have expression because there's nothing there because something's been induced to leave, right? Or do we have something there because there's no repressor present, right? So nothing is there to repress transcription, right? So it's really kind of what's the end goal and then what has to happen for that end goal to be achieved. And it's funny because I always have to think very clearly about these in this very same way, right, that I'm teaching you. So I don't tie myself in knots, basically. And so repressible transcription, default state is on, right? So there's nothing there. So this is very much applicable to prokaryotes only. Well, yeah, I always got to be a little bit careful about making definitive statements. And so in this case, we need a repressor to bind. And that repressor is a two-part deal, right? So in this case, we have in repressible consists of two parts. Typically a protein that can bind DNA, but will only do so when a co-repressor is bound. So no co-repressor, no DNA binding, the repressor does not act as a repressor. So as you can see here, in inducible transcription, it's the presence or absence of the inducer that matters. In repressible transcription, it's the presence or absence of the co-repressor that matters, right? So here are switches, right? Here are ways of flicking on or flicking off transcription, right? Depending on the presence of something, whatever that something may be. Quite often in prokaryotes, it's a metabolite. So just to put that into kind of pictorial form, which always helps, in inducible transcription, transcription is off because a repressor is bound to the operators, typically what we call it in prokaryotes, and you don't get any transcription. Only when an inducer is present does that repressor leave, right? And now we have, you know, the bare DNA, and in prokaryotes, that equals transcription. Right, so you can see how these are very much terms that work best with prokaryotes. Now, in repressible transcription, you have this protein present all the time, but in the absence of the co-repressor, this protein cannot bind DNA, right? Its shape is wrong. Essentially, the co-repressor changes its shape. Actually, in many ways, in the same way as inducer changes the shape of this repressor, right? It's all about changing the conformation of the protein. So inducible transcription, in the absence of a inducer, has the correct shape. Inducer is present, has the wrong shape, and can no longer bind DNA. In repressible transcription, it's the opposite. In the absence of the co-repressor, that protein is present but has the wrong shape, right? Can't physically bind DNA. Only when the co-repressor is bound to it does it change its shape, and now it can physically recognize the DNA and block transcription. right? So these are kind of opposite sides of the same coin in many ways, right? In inducible transcription, the protein will bind in the absence of inducer and is then essentially removed or not allowed to rebind because this is a dynamic process. And so transcription is on. In irrepressible, in the absence of the co-repressor, transcription is on by default. But when the co-repressor is present, now the repressor takes its active form and stops transcription. How did that go? I want to make sure that this all kind of clicks, because without understanding these principles, the rest is very difficult to get straight in your head. Okay, just going to go on to the next slide and then I'm going to call it a day so we can get out in time. So obviously it's not as simple as this. I already told you the story about the Gene Reg Journal Club at Penn State that I was strongly recommended not to join because it is crazy complicated. So you can have both aspects. Obviously, as you get to eukaryotes, things get way crazily complicated. But even in prokaryotes, you can have both positive and negative regulation, right? Typically, negative regulation is more common in prokaryotes, and positive more common in eukaryotes, right? But those aren't like hard and fast rules because it really depends on what kind of level you're talking about, particularly in eukaryotes. And also, many genes, especially in prokaryotes, exhibit something called autoregulation, right? So the product of gene expression, whatever that produces, its product can then regulate the transcription of the gene that produces that protein. And so the easiest way to think about this is in terms of metabolites or metabolism. And so essentially, once you've produced enough of something, whatever metabolite that is, you don't need as much gene expression to maintain that level. And so you then want to dial back gene expression so you don't produce an excess, right? It's kind of like homeostasis, maintaining the correct balance. All right, I think that's enough for today. If you can, scan through this PowerPoint, right? There's a lot of stuff going on. And particularly when we get into the lac operon is so-so. But the trip operon is really challenging, right? So having a heads up of that will help you a great deal. Okay, I'm going to grab a coffee and then head out. So see you all later, tomorrow or Wednesday, and keep reading up on this stuff in the meantime. It will help you a great deal. Cool. All right. Have a great day, everybody. It's a beautiful day out there. Hope you get to go outside and see you soon.